Our next speaker is Dr. Robert Hampson, who is an associate professor of physiology and pharmacology at Wake Forest School of Medicine in North Carolina. His dedication to science advocacy and brain awareness has led him to pursue these interests as an advisor to science fiction authors and to represent neuroscience and biology at many science fiction conventions. We've crossed paths many times. I'm never sure what name to call him. He has to have a different name when he goes to fandom. He might want to explain that at a break sometime. But he is well published with over 100 peer-reviewed articles, 20 book chapters, 2 patents, and 30 years of presentation and teaching. So I'll turn it over to Dr. Hampson. Very good.
Thanks for coming. Okay, thanks. When Les suggested I might wish to submit a proposal to speak, I kind of wondered what I could talk to this group about. And then I realized that there's a number of things we really need to understand about the brain before we go. Everything that's been talked about with understanding the ships, how are we going to get there, what are the propulsion, what are the habitats, but what about the human body before we go? A little bit came out in a question I asked earlier. I do need to say that for the past 30 years, I've been working as a physiologist and pharmacologist. My work has ranged from drugs of abuse to cognitive enhancers, from sleep deprivation to PTSD, and more recently in the field of neural prosthetics. Now, because this says Wake Forest here, I have to give the caveat that this is my position and not anything official from my institute. However, they do like the fact that I'm out here promoting brain awareness and talking to you guys a little bit about what the field is. Today I'm going to touch on the idea of neural prosthetics, and I will mention with Tony in the audience that there's some unknown fellow by the name of Ted Roberts that's going to have an article on the Bain website here coming up fairly soon that's going to talk about neural implants and interfacing the brain. And yes, Les, I do have to use a different name every time I go somewhere. But let's get our presentation here. Come on. There we go. Back up. So let's talk a little bit about what do we need to know about the brain before we go. So I'm not going to talk about psychology. I understand Sam is actually going to talk a little bit more about psychology, but I'm going to talk about what do we need to know about the brain in particular. We need to know about the physiological challenges of space and microgravity. We heard a little bit about that this morning, and we need to deal with the duration of interstellar travel. We just heard 50 to 100 years. And we also want to consider that one of the best ways to reduce physical risk is not with robots, but with teleoperation. So for teleoperation, we're going to need to know a lot more about the brain, because if you're sitting there working Waldos and you're working remote manipulators, it's not very efficient. So the best way to do this is going to be with some form of a direct neural interface. So how do we get there? So since I know that this is not a brain scientist audience, this is a brain. And I've listed a number of the primary areas. Essentially, vision is here, sensory information of the body's here, motor control of the body's here, memory and hearing, and decision making. Each of these brain areas has a strength with respect to what we can use it for, how we can interface it, either with a brain-to-computer interface, a brain-to-machine interface, with a prosthetic or teleoperation. But I also have several very key weaknesses. And I've listed pluses and minuses. Parietal lobe, one of the pluses to interfacing it is machine senses. One of the minuses is that it's very radiation sensitive. Occipital lobe, vision, artificial vision, but also sensitive to traumatic injury. And my favorite here, temporal lobe, there's a small structure about an inch in from your temple called the hippocampus. The hippocampus is the part of the brain that processes new memory. And the hippocampus is very prone to epilepsy. It's also prone to stroke. One of the issues that's going to come up is if we launch 100 people in a probe 50 to 100 years, 25 of those people will be incapacitated by the time they arrive. They will become incapacitated unless we can better predict the onset and occurrence of Alzheimer's disease, the onset and occurrence of epilepsy, the onset and occurrence of stroke. And as long as and just understanding what microgravity effects on the heart and bone are not enough. We need to know how it affects the cerebral blood flow, the cerebral circulation. So we need a better understanding of radiation effects on the brain. We need better understanding of microgravity effects on the brain. And we need a way to predict and prevent degenerative diseases. The illustration I'm using right here of Alzheimer's disease shows that what we see with Alzheimer's disease is a progressive degeneration that is caused by a buildup of inert material in the brain, which essentially, neuroscientists don't like to be inaccurate, but the analogy is it chokes the brain cells. When you have anywhere you have a tissue that is very highly metabolically, chemically, electrically active, if you add inert protein to it, it's not going to be as efficient, it's not going to be as active. And to keep the brain healthy, we need to consider entertainment and intellectual stimulation. So, okay, maybe this is a little bit about psychology, but one of the ideas is, can we do this with direct interfacing? And I had a specific question asking me, how long before we can have our TiVo directly wired into the brain? How long is it now until our Halo 7 is done with a direct neural interface? Xbox 360. Another question, hibernation. Can we put the body and the brain to sleep? And if we do we have to do something special? Do we know if a hibernation effect is actually going to suspend brain activity? And what's the consequence if we do suspend brain activity? Because right now as it stands, your brain is active 24/7. You may not think about it, you dream, but dreaming is just one stage of what goes on during sleep. And if we hibernate our astronauts, are they going to be one continuous dream for 50 years? That is one of those things that we don't know. So this is another aspect of things we need to know about the brain before we go. Another one is autodocs, or any of your various science fiction catchwords for an automated medical support unit. Something that can perform the work of a doctor, including doing surgery, microsurgery, or anything of the sort. If we had Star Trek transporters, it would be perfect. We can go in there and we transport the inactive or the diseased portion of the brain. If you develop a blood clot that is right on the motor cortex, and you had some way of going in and removing it, what does that require? Nanomachines? Microsurgery? Transporters? Teleporters? If we have a way to fix the neurodegenerative disorders when they occur or even before they occur, then now we start improving those odds of arriving at Barnard Star or Alpha Centauri A and B with 90% of our original population instead of 75%. The brain is both chemical and electrical. It's quite frequently you will see claims for parallel computers that say we have computers with enough processors as there are neurons in the cat brain or in the monkey brain. Therefore, we have a computer smart as a cat, as a monkey. That is not the case. There are approximately 800 trillion neurons. Each of those neurons has a minimum of 1,000 synaptic connections. It is not the number of neurons that we need to worry about, it's the number of synapses we need to worry about. So we've just added 3 orders of magnitude to anything we might want to do to try to imitate that, and it's an electrochemical device. The chemical is that every neuron that interfaces, connects with another neuron, releases a chemical that causes channels to open. Through those channels, we have sodium chloride, potassium chloride, calcium, magnesium, flowing through those channels, and what they do is they're all charged ions. And those charged ions then cause a buildup and then a release of electrical voltage, electrical potential. So now if we're talking about a bus or dramjet with a large, heavy magnetic field, or we have a fusion reactor that might have a leaky electromagnetic field, what's that doing to the brain? We frankly don't know. We do have a clue. And one of those clues is based on what I was asking Dr. Davis earlier, which is radiation effects on the brain. We know now that patients who have had to have gamma knife or whole brain irradiation within 6 to 12 months start losing their memory. And this is a real problem. And the problem, in fact, is one in which the radiation causes heating, the heating causes inflammation, the inflammation causes cell death. And if we can find a way to block that process, which is another project that I'm loosely affiliated with, then perhaps we can avoid or eliminate some of this damage. So how do we connect to the brain? As a matter of fact, most of this audience is old enough to recognize $6 Million Man. Okay? And $6 Million Man was fantastic. It was the motivation for getting me into this field. It only took 25 years to actually invent bionics. I started off and I said, I want to do bionics. And immediately found out that there was no such field. There is a field now. Actually, we call it neuroprosthetics. We don't call it bionics because bionics is a trademarked name by a company for a muscle stimulator that is not really what we would consider from $6 Million Man. But how do we connect to the brain? Prosthetics, exoskeletons. Wouldn't that be great? You end up on a world that has 1.5 times G, and you don't have to worry about the gravity load because you've got an exoskeleton. Well, DARPA does have an exoskeleton, and the power supply is about the size of a truck. Brain-computer interfaces. Again, if you could, to prepare your colony site, if you could leave your technicians in orbit while they're teleoperating machines on the surface to prepare the area, then you gain an advantage of not exposing your astronauts and your colonists to the hazardous environment immediately. Eventually they will get there. So how do we connect to the brain? Well, here's the state of the art. I'm going to show you a video. I'm actually going to switch to the long video here. Come on. And this comes out of University of Pittsburgh. I know the principal investigator on the project. I contacted him prior to this, and he steered me to the appropriate videos and then gave me a few words of caution, which I will add once this is over. Oh, by the way, first thing I want to call your attention to, I'm not quite sure how clear it is, but you're going to see 2 permanently embedded contacts right here. And those permanently embedded contacts right now are the only way we have of interfacing with the brain. Okay, I will cut that because there's much more. In the interest of time and staying on time, I'm going to cut. I had another video I was going to show you. I'm actually been told for IP reasons I can't show. But this is what I call an output prosthetic. Input prosthetics bring senses into the brain. Input prosthetics would be cochlear implant, retinal implant. And as a matter of fact, right now, we don't have implants that go straight to brain. The only thing we do is tap into the remaining neurons that are left in the cochlea for hearing or on the retina for vision, and find a way to get a signal to those, and then that processes the signal and sends it to the brain. This is an output prosthetic. An output prosthetic is now there is an implant, and that is 100 electrical contacts placed right on the motor cortex of the brain, the driving area for controlling the hand and for the arm. She's actually got 2 of them in place, one for the hand and one for the arm. And then there's another type of prosthetic that I call a brain-to-brain prosthetic or a processing prosthetic, and that's one in which what you're trying to do is not an input, not an output, but bridge a damaged area of the brain. That happens to be what I'm working on. Maybe next time we do something like this, I can tell you a little bit more about it. We're making some incredible steps and working again with human patients. We've had quite a bit of success with animal models, and we're trying to take this into humans. But what we need right now are two major computational problems in neuroscience. One is what we call the direct problem. The direct problem is you take firing of single brain cells, single neurons, and use that. That's what's going on with this patient. And by the way, there's only 10 neurons that are doing most of the driving. And yet there's thousands in this area of the brain that are active, but only 10 neurons are necessary for driving this. And Andy Schwartz, the professor who's behind most of the research here, cautions me to say that we really need to be very cautious. We don't really understand the brain. We've just figured out how to make certain things work. And what's really happened is we've taught this patient, and she's using the plasticity, the flexibility of her brain in order to make this work. The direct problem then is we can figure out what the effect of one neuron is on EEG, electroencephalogram, the recording from brain surface or from scalp surface. The inverse problem is being able to predict those single neurons from the EEG. And we don't have a way to do that. Here's a measure EEG from surface electrodes, and we have these continuous waveforms. But when we're looking at single neurons, we're looking at something that looks much more like this. And there is a portion of the video, and anybody who asks me, I'll tell you where to go find these videos. There's one that shows, okay, think about raising your arm, and all of a sudden you hear pop, 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 as they're recording from a single neuron. Okay, now stop. And it goes silent, because she has control over the neural activity of her brain. What we would really like to be able to do is do all of this without those implants. But what we need to do it is we need the help of the people in this room. In order to preserve or fix the brain for a long duration space flight, we need to be able to interface with it. To interface with it, we need to understand the language of the brain, and to understand the language of the brain codes, we need to do it non-invasively, and we need to solve the inverse problem. The inverse problem is one that can be solved with mathematics. But right now, just trying to figure out the activity of 10 neurons requires a supercomputer. And there are, at last count, approximately 64 million coefficients in this model, which is, unfortunately, not showing up. It's in a bad, let's see if I can get this to zoom. Okay, this is a Volterra model, nonlinear, and you can see loads and loads of sigma signs. And this is just a linear approximation of a nonlinear model. This is not the full model. But this is what we need help with. And this is where we need to go. So from here, we go to, I'm sure you've seen some of the pictures that equate star fields with neural fields. And if we can figure out the interfacing to the brain, then a number of the problems that we do face with respect to getting humans out into interstellar space can be solvable. But as I said, this is one of the reasons why I came here and decided that if I can enlist your help, then you can count on ours. And thank you very much.
We probably have time for a question or two, and that's it. So.
You want to call him? Oh, okay.
How much does microgravity affect synaptic geometry and the way that the brain processes?
We have no idea. Last night, you asked me a question about setting up a biomedical laboratory in space. Right now on Space Lab, we've flown maybe a total of 100 mice. We need millions of mice, thousands of rats, hundreds of dogs, and hundreds of monkeys in a biomedical laboratory in space to start figuring this out. We need approximately 50 million cubic meters of space to build a laboratory. Yeah.
You alluded to brain augmentation early on in your talk. Nietzsche referred to human beings as a bridge, or more specifically a rope between animals and superhumans. And I want to ask you, this is specifically quite a provocative question, but at what level of augmentation do you cease to be Homo sapiens, in your opinion?
When there's no human in the loop. As long as there's a human in the loop, you're still humans, Homo sapiens. Yeah.
This will be the last question.
You're talking about the direct contact was the only way that you could access the brain for the purposes of her prosthetic arm. Have you guys tried anything like with a Bluetooth remote technology to make that work?
Well, yes, but see, we still have to have the wires in the brain. We can have the Bluetooth. In fact, that's one of the two patents that Les mentioned. But we can have Bluetooth connectivity, but we still have to have wires in the brain. And
You're just porting out to the Bluetooth.
right. Now, that video was from a 60 Minutes segment a couple of months ago. It's about 13 minutes long, and they do show some work with a strap-on robotic arm that is powered by the surface pickup from the stump of an amputee. And there are ways to pick up some signals from the surface, but if you're talking about augmentation, or if you're talking about a prosthetic to treat Alzheimer's disease, that's not going to work.
Oh, no, I didn't want to be unplugged. Okay.
You'll need to run it around the other side. Thank you very much.